Good evening, everyone. A warm welcome to all the participants and others who will keep on joining later. So, I welcome you all on behalf of the optical sensing and monitoring community. So, in this webinar series, we'll have Dr. Federico Formenti, who will be speaking on shining light on oxygen, preclinical application in blood and gas. So, before formally introducing Dr. Federico, I'll just for a minute, I'll brief you about the optical sensing and monitoring group. So the purpose of this community is to basically support the reach of sensors from research to end users and also have a common platform for interaction among various stakeholders from industry, academia, end users, the funding agencies, and all bring together on a common platform to discuss the issues. Also, the current activities which are ongoing, so there are like I would have generalized over here like 4 type of webinar series. One is the journey, so how sensor has progressed from a very basic lab work to end work or at a very clinical application stage. Other is collaboration, because all the sensor development depends on collaboration. So different groups from different backgrounds coming together. Then sensing needs, so bringing people from agencies or pollution control bodies where they can say what are the sensing needs where researchers can work on for future, and learning talks where people come and explain some concepts which we can learn. Also, there are symposium work which are going on, so I will you'll get some more details later on. Also, there's work on developing various technical articles and information sharing. And the work in progress is the main website of the community, and there's an optical tinkering facility. So this is work in progress, which is going on in the background, and hopefully you'll get more updates. I'll also put in the chat the webpage, LinkedIn webpage, and you can connect over there for future updates. And before going ahead, I would like to acknowledge, so this optical sensing and monitoring, whatever background work is happening with tinkering facility and all, is by support of the Optical Society of America Foundation. So last year I got this fellowship, Milton and Rosalind Chang Fellowship. So based on that, I'm working on that. Also for today's talk, I would like to thank Professor Soumya Mukherjee, Mr. Hitesh Mehta of Eagle Photonics for communicating to the Photonics India group, Mr. Mukesh Vishal for helping with the platform. With this brief, I would I would like to welcome today's speaker, Dr. Federico. So I'll just give a brief introduction of him. Dr. Federico Formenti is a physiologist and sports scientist. He obtained a doctorate in philosophy and human physiology from University of Oxford in 2011 under the supervision of Professor Peter Robbins and Keith Dorrington, and a PhD in human biomechanics from Manchester Metropolitan University in 2006. He worked as a postdoctoral fellow in Division of Anesthetics at University of Oxford until 2016 under supervision of Professor Andre Farmery and Clive Han. He is currently a senior lecturer in human physiology in the Center for Human and Applied Physiological Sciences and honorary senior research fellow in Division of Anesthetic, Newfield Department of Clinical Neurosciences, University of Oxford, and also an adjunct associate professor in Department of Biomechanics at the University of Nebraska in Omaha. And he's also a fellow of Higher Education Academy, a fellow of Physiological Society, and a Kelly Visiting Fellow at Wadham College, University of Oxford. He's also senior editor for Experimental Physiology, a reviewing editor for Frontiers Medical Technology, and an ad hoc reviewer for several journals. His research interests include human response to hypoxia, cardiopulmonary, and exercise physiology, development of technology for physiological reach, and critical care. And today he will be presenting his work. So this talk falls into the webinar series. I would say this is a combination of journey and collaboration, how they started the work almost a decade back, and how it has progressed to a preclinical stage, and further they are working towards clinical applications. And with that formal introduction, I would just like to say, so in spite of all that things, he is a very humble and very kind-hearted person, and he's been very kind to be able to, even though he has a very busy schedule, he has been kind enough to take out time and volunteer to share his work to the community. So with that, I would like to welcome Dr. Federico to come on the platform and take over the session.
Thank you, Nirmal. You've been too generous. You've said too many, too nice things. It's me who has the pleasure to present some of our work here today. So thank you very much for this opportunity. I'm glad to have the opportunity to share quite a lot of our work with you today. Let me see if I managed to share my screen, and if you could confirm whether you can see
your screen is visible.
the slide. Fantastic.
Yes.
Thank you. So yes, what I'll be talking about today is the preclinical applications in blood and gas of some fiber optic technology that is used to sense the partial pressure of oxygen. The figures here show a little bit of an overview of some results from bench testing in gas, some results from preclinical work looking at the clotting resistance of these fiber optic sensors, some other tests in the animals in terms of matching between measurements done with a sensor and standard blood gas analysis. And then some imaging here. These are the lungs of a pig, and how we transferred the use of this sensor from an application in arterial blood into an application in gas, looking at what happens down the airways in a pig's lung. But just so that I'm clear from the beginning, let me see if I can get on to my next slide. What I'll be talking about today is not quite my own work. I have done my part, I think, but I'd like to acknowledge, first of all the colleagues that have helped doing the experiments, planning them, the analysis, and presenting the results that are listed here on the right, and the funding bodies that are presented on the left. So I've been truly blessed to work with amazing supervisors and amazing PhD students or master students that really have enabled all this work to be done. My talk is going to be divided into 4 sections, not necessarily of equal time and work. I'll introduce the topic by mentioning the importance and the clinical needs. So why should we care about fiber optic technology in the clinical context of critical care? The second part will be a brief overview of the
Dr. Federico, your mic is on mute. I think we lost you somewhere in between.
Yeah, sorry. I saw the host has muted everyone. But let me try to reshare my screen again. If I do, it all went back, I think. Chip. And could you confirm that you can see the slide here again?
Yes, we can see these slides.
Excellent. And Nirmal, where did I get muted or disconnected, please?
In the initial part only, you got muted.
Okay, fantastic. So yes, I was saying that initial part of manufacturing was led by my colleague, Dr. Rongsheng Chen, and the company that has worked with us for more than 10 years now, Oxford Optronics, and I'll present a few results from bench testing. The larger part of the talk will be on the preclinical studies and a brief mention to ongoing translation. And here we looked at the agreement with gold standard blood gas analysis, and we tested the rapid response in some animals before we move on to studies in patients. And in the end, I'll conclude briefly with a mention to other applications. I'll mention something about lung surgery for patients with lung cancer, for example, where again, we've done the preclinical study and we're hoping to move to a clinical work soon. An example that applies to equine anesthesia, these are unpublished results, just one slide on skeletal muscle, which is work done by an excellent group in the US. So first of all, why would we need a fiber optic oxygen sensor in the critical care unit? So there is a need for personalized mechanical ventilation for patients with a condition called acute respiratory distress syndrome in the intensive care unit. These patients are hypoxic, and it is an inflammatory condition. And you see here on the right, there's this cartoon taken from the New England Journal of Medicine, and there's this patient that is being mechanically ventilated. And there's two physicians that are staring at the patient monitor and the ventilator. And one of them says this patient is in ARDS, so has this critical care condition. And they're wondering how they should adjust the mechanical ventilation. These abbreviations say that they could alter the frequency of the breaths. So say they could give more or fewer breaths per minute. VT is the tidal volume, is the volume of gas that goes into the patient at each breath, and some different pressures here. So while there are some general guidelines that come from large clinical trials and they work on a population level, there is not currently a bedside technology that is available to monitor what is called pulmonary atelectasis continuously in real time. That means that there's nothing really available at the bedside that can tell us in real time if the ventilator settings we're using for that particular patient are appropriate or are not. We know that they would work on a population level, but are they appropriate for that individual patient? We're not so sure. In the context of COVID, this is a picture taken from one of our colleagues' publications last year, where we see that the same patient over different weeks showed very different lungs. This is a slice of the thorax of the chest of the same patient, where black is air, like outside of the body. You see it's all black, so that's air. And inside the lungs, there was lots of air at this point in time. And later on, there was not as much air. You see that there's some consolidation, or it's called atelectasis, in the lower part of the lungs in this picture. And you see that the arterial oxygen levels, while the two lungs were very different on these CT scans, the arterial oxygen levels were actually not 4 millimeters of mercury. So putting things into context, you see here that there would be a need for a system, a device, an approach to tell at the bedside, without taking this patient away from the unit into the CT scanner, to say, look, yes, the arterial oxygenation is about the same, but perhaps in one of the two, the continuous recording would look very different, has a different pattern compared with the other scenario. So one of the key aims of our research is to use fast responding fiber optic sensor to detect arterial oxygen tension, respiratory oscillations in these patients, and the association with mechanical ventilation. And I'll show you how we propose to do that. There's the rationale behind our work is based on something called cyclical atelectasis. Cyclical because it works in cycles, like the respiratory cycle when we breathe in and out. And atelectasis is the collapse of lungs regions or units. So this is really the alveolar opening and collapse. These two videos come from a paper published more than 20 years ago now. The reference is in the bottom left corner of the slide. And they show an excised lung of an animal. This has been taken out of the chest, so it's not the same as inside the chest. But it shows some pink regions, which is where the lung is collapsed, and some lighter pink or white regions, which is where the alveoli are open, the air units, the little sacs in the lung are open, and then there's air inside here. And we will see in a moment what happens when air is delivered into this lung under pressure, and we'll see it popping open. This is some fluoroscopy, so it's as if we were able to put a small camera somewhere, for example, here where it's reddish or dark pink, and we'll see what happens when air pressure is increased down that pipe. So that is visualized in these two videos, and you see that when pressure is applied, the lung opens up a bit at a time. Some parts become white straight away, other parts take longer, but if that positive pressure above atmospheric pressure is applied for a given period of time, we see that most of the dark red regions can be so-called recruited, so they can be filled up with gas. And similarly, here in the fluoroscopy, we see that at the zooming into, say, one of the lung regions. Now, if this is correct, how do we transfer that concept that we've seen just one event of positive pressure, which is applied for a long period of time? That's obviously different from our normal breathing and from conventional mechanical ventilation. Well, we see here, this is a study published also quite a few years back by Professor Tobin. And you see that with conventional ventilation, in a period of 6 seconds, the mechanical ventilator could deliver, for example, 2 large breaths, more than 800 milliliters. And what could happen in this case is that if we allow all the air to come out of the lung, there would be only a limited amount of air in the lung at the end of the breath. But if we don't maintain the pressure in the lung above atmospheric pressure, it is possible that some alveoli collapse at the end of expiration. Then they may open up, as in the video we showed a moment ago, during inspiration. But if we deliver an excessively large breath, it may be that we're stretching these alveoli beyond their capacity, and that alternation between stretching them too much and allowing them to collapse is known to be dangerous for the lung. So more recently, a so-called protective ventilation is being used, where you see that in the same period of time, smaller breaths are delivered, possibly around 400 milliliters. That is half of the tidal volume we had earlier. And as a consequence, to deliver the same amount of air in a minute, we must double the number of times for which that breath is delivered. So we have breaths which are half the size and twice as many in a minute. And you see that that volume doesn't go to zero, and that is because it's illustrating that we let more air into the lung at the end of expiration. So the pressure in the lung is above atmospheric pressure. By doing that, we avoid the collapse of the alveoli, or we limit it. And so if the alveoli remain open, gas exchange can continue. If we have a collapsed alveolus here, there's no gas inside the alveolus, and so it is impossible for oxygen to be taken up by the little blood vessels around the lung. In contrast, if we always maintain some gas into the alveolus, the pulmonary blood vessels that go by that alveolus will be able to take up oxygen and release carbon dioxide. Now, one of the first observations in terms of the associations between mechanical ventilation and ventilatory oscillations in the partial pressure of arterial oxygen was published in 2002 by an excellent group with Dr. Baumgartner and Professor Mark Staller's groups. And it was an experiment done in rabbits, small rabbits. And you see here over a period of time, these are seconds on the horizontal axis, and we have the partial pressure of arterial oxygen on the vertical axis. What changes between the left part of the figure and right is RR, which is the respiratory rate, where 20 breaths per minute were delivered in the first part of the figure and only 10 in the second. And you see that the size of these oscillations pretty much resembles the size of the oscillations in the figures on the right, meaning being interpreted as a lack of cyclical atelectasis when we have an elevated respiratory rate, which could happen during a reduced respiratory rate. So it is possible possible, based on this arterial oxygen partial pressure signal, that having an elevated respiratory rate maintains the lung units open and allows for gas exchange to continue. In contrast, if larger breaths are delivered and fewer per minute, there's these large swings between oxygen that increases during inspiration and drops very rapidly during expiration, possibly because of this opening and collapsing of alveoli in the lung. So how would the arterial oxygen partial pressure signal help in managing mechanical ventilation? This figure that Andrew Farmery put together for a review published in Anaesthesia shows the context. We see here the partial pressure of oxygen signal recorded continuously, which can be an input similar to what could happen for electrical impedance tomography and imaging technology, into a computational model which would receive inputs also from standard monitoring that is available in mechanically ventilated patients. For example, the monitoring of the tidal volume, the volume of air that is delivered per breath, and the pressure in the airways. All this information, together with this arterial oxygen signal, could be used by a computational model, which would be used to determine, as illustrated by the dashed line, the settings of the mechanical ventilator. For example, the pressures that are necessary for ventilating a given individual patient, or the respiratory frequency, how many breaths we could give per minute. Now, so where do we go from here? How do we put together a sensor that can give us that signal, which itself can help guiding mechanical ventilation? So the initial stages of development here were in terms of manufacturing. That was led largely by Rongsheng Chen, together with Trevor at Optronics, who were and are manufacturing the sensors. What we needed is listed here at the bottom, is a small sensor to fit into a standard arterial catheter that is illustrated in the photo on the left. It must be made with medical grade materials. The rabbit experiments I mentioned earlier were performed with a sensor which is made with ruthenium. And while it is fit for purpose for research reasons, it is toxic for use in patients. So that doesn't enable the translation to the clinical settings. The sensor needs to have a very rapid response time, because if we take a breath every 3 seconds, it must be able to respond within those 3 seconds to show us a respiratory oscillation. And also it must be resistant to clotting, because if a blood clot develops on the sensing tip of the fiber, that clot would increase the distance between the medium, in our case the blood, and the fiber, hence increasing the response time. So if we have a rapid response time but no resistance to clotting, it may be that we get the signal for the first 5 minutes, for example, but then that signal wouldn't be responding sufficiently rapidly later on due to the clotting. And the two pictures here are just to show you what the sensor looks like. I'm sure you're all more familiar than me with fiber optic technology here, and some of the dimensions. And this is an early version, actually, of the sensor, where Oxford Optronics has now implemented an EEPROM with calibration values that are embedded into the sensor, so it becomes a plug-and-play device. In terms of the working principle, this illustration from Rongsheng's paper in 2014 shows the components of the system. This is the fiber optic itself. The sensing part is at the tip. We have a source of light, an LED here, where the light enters the fiber via a fiber coupler, and then the sensing part of the fiber is illustrated here at the top, where we have a blue arrows that indicate the light coming from the LED source, and that bounces around into the tip of the sensor. The red arrows indicate the interaction with the oxygen molecules, and then the red arrows that return into the core of the fiber illustrate the signal, which comes back to the fiber coupler and enters the spectrophotometer. And the intensity of the light was what was measured as a signal at the time, and then converted to partial pressure of oxygen's values. And at the beginning, one of the key steps in the development was investigating molecular structures of polymer matrices and their response time. That was initially done in gas, in that it was much simpler on an experimental level. And the key point of the rationale here we see some of the polymer matrices that Rongsheng tested and the associated response time in the graph over here on the right, where you see that the 3 different matrices had different response times. And in this example here, the PPMA matrix had the fastest response time, as in response to a square wave change from 5 to 15 kilopascals in terms of the partial pressure of oxygen. You see that the vast majority of that change was detected within a fraction of a second. Here we have time in seconds from 8 to 9 seconds. And in my simple mind, the way to understand the functionality or the functional significance of these matrices is by looking at what happens if we have different size of stones and sand, and we want to filter and we want to get water through them. The long chains of pendants here in PPMA are illustrated by gravel, like large stones. And in contrast, the PMMA matrix is maybe illustrated here by sand or silt. And you see that in 2 minutes in this analogy, if we pour water onto gravel over a height of 1 meter, the illustration shows that a lot of water can make it through the gravel in 2 minutes. In contrast, if we fill up the same volume with sand and we pour the same amount of water into the box through sand, after 20 minutes, there's only a little bit of water that comes out. And that is the analogy in my simple mind with oxygen that can enter them and leave the matrix very rapidly. If we have longer chain pendants in PPMA, it is possible for oxygen molecules to diffuse and leave the matrix very rapidly, in contrast with what would happen, for example, here with the PMMA matrix, which has a slower response time, and the analogies with sand or something similar. Now, there's been lots of bench testing done by Rongsheng, even before I joined the group and after I joined the group. I've only thought I'd show the result from one key feature of the sensor, which is the fast response time. There are similar sensors that measure the same thing, but they cannot, they don't typically show the fast response that these sensors have. And here the experiment was done in gas, and again you see a very rapid response. In this case, all matrices showed a reasonably rapid response. Relatively speaking, the PPMA was the fastest. And then, as per title of the webinar, I'll focus a bit more on the preclinical studies. I'll mention about the ongoing translation. I imagine many of you may or may not have experience of preclinical studies. This picture was taken in Uppsala. This is the CT scanner, the little donut. This is Nikhil Bomakanti and Anders Nordgren, who were helping at the time. And I thought I'd just show you the picture to have a feel for the complexity of these experiments. There's lots of monitoring, the same as probably would be used for a patient. All the anesthesia kit, obviously the top priority is for the animal to be comfortable and without pain. And the mechanical ventilator that we used, the same as would be used with patients in intensive care unit. And so it's a rather challenging logistics, and there's lots of kit that needs to be collected. It requires quite a few people to do the experiments, and the data analysis is reasonably substantial. Now, some of the experiments we did at the beginning in terms of the resistance to clotting were done at the University in Pilsen in the Czech Republic with Martin Matejovic's group. And here, one of the first upgrades from the bench testing was that we could put the sensor in animals that were used for other experiments. We were not interfering with them, but we could put the sensors in these animals for periods of more than 24 hours. And the blood of these animals was just as it is naturally, so it didn't have any drugs that would avoid its clotting. We had to use anti-clotting drugs in the bench testing, because otherwise on the bench, when we used blood from animals or donated, the blood would clot into our circuits, and then we couldn't run the experiment. So we could test the response time on the bench in blood, but it was blood that would not clot because of the drugs we used. Whereas in these experiments, we put the sensors in arteries and veins for more than 24 hours, and then we looked at them with microscopy and with electron microscopy, and we measured in a way we weren't able to measure any biological material deposition on the fiber, suggesting or indicated that they were resistant to clotting. And then obviously we repeated what was done on the bench. We looked at the accuracy of these measurements. The graph here towards the middle shows a period of 35 minutes on the horizontal axis. And on the vertical axis on the right is the fraction of inspired oxygen. So the animal at this point would have been given 100% oxygen, or at the beginning of the experiment would be given room air oxygen, which is about 21%. And on the vertical axis, we see the partial pressure of oxygen, where we have two signals here. We have some green dots, which are a standard blood gas sample. So it's taken with a syringe, as is normally done in patients. And then together with that signal, we have in blue the continuously recorded signal, which was recorded with our sensor. And you see that there's reasonably a good matching between the two measurements, and the agreement between the two measurements with standard blood gases, ABG, and our sensor is demonstrated on the right with this Bland-Altman analysis. This new technology also allowed the study of cardiopulmonary physiology in more detail in a way that had not been done before. That is a separate part of the topic, which I'm not going to focus on today. But closing the circle here, one of the key points was actually measuring ventilatory oscillations in an animal before we can move on to patients. And here on the right, we see two graphs with periods of time of about 30 seconds on the horizontal axis and pressures on the vertical axis. Now, the arterial partial pressure of oxygen that was measured with our sensor is illustrated by the black lines at the top. And the other two gray lines going down are the pulmonary artery pressure and the airway pressure. They essentially indicate, for what we're interested in here now, is when each breath was delivered and the way in which it was delivered. So here there was a constant flow of air into the lung. It is called volume control. And on the right is called pressure control. And you see that instead of a constant flow, there's a very rapid flow increase at the beginning of inspiration, and then a pressure is maintained for the duration of inspiration, and then it is released as is done during expiration. So at this stage, we were able to essentially demonstrate that the sensor responded rapidly, was able to track ventilatory oscillations in animals, and didn't clot. So we thought that we had sufficient evidence to move on to patient studies. In this sense, the translation as you see here, is taking place in two steps. One, which is what I call is the external sensor project. It's led by John Cronin and Luigi Camporota and funded by Barima and the National Institute of Academic Anaesthetists in the UK. And you may well tell me, Federico, there's a mismatch between the patients in the title and I can still see a pig in the picture, and that's correct. What we've done so far is the measurements in animals. This device here is an aspirator, which is electronically operated, and it's connected with the syringe that draws blood out at a constant flow out of, in our experiments for now, a pig's artery. And you see the fiber is the white line here, which goes into this Y-shaped connector. It's not inside the pig's artery, but it's just outside the pig's artery. So this approach would have relatively limited ethical implications. And you see on the right, there's a little video that shows an operator opening the tap, which is that white tap in the picture on the left, and that allows the drawing of blood into the syringe, and that blood flows onto the fiber to measure the partial pressure of oxygen. These experiments have been delayed by COVID inevitably, but they should take place this coming year. And the other larger study we have funds for is the one where we take this sensor into patients' arteries. It's what I call the first in-man measurement. And this study is funded by the NIHR in the UK, together with Professor Andrew Farmery. And again, pending the COVID delay, we're hoping that next year we'll be able to do the first in-man measurements of arterial partial pressure of oxygen in patients in the intensive care unit. And finally, I'll conclude with mentioning some of these other applications. I'm quite excited about the lung surgery one, in that it is a study that we performed in the past year, not more. And I think it shows an entirely different application of the same technology. So here, this work was very much done by Jack Galiphant and John Cronin. I was just helping them getting through things, but they're the ones who actually did the work. And essentially the context here is the one of patients who may have conditions in their lungs, for example, lung cancer, where it may be that one region, one portion of the lung is entirely affected by cancer, and other regions are not at all. And in that case, the current approach in terms of determining whether that patient may or may not benefit from lung surgery are based on measurements that are illustrated here. And these measurements don't look at the lung as a system made of different units, but they look as a lung as a single compartment thing. There's like a one box rather than one box made of different smaller boxes. So for example, we see on the left, there's an individual who's doing an exercise test on possibly a treadmill. And on the right, you see one of the typical respiratory maneuvers where a patient takes in a deep breath and then blows out as hard as possible into a tube. And then the machine records the results of this so-called spirometry test. And we're looking at how, say, air comes out, how much air comes out in, say, the first second of expiration. And obviously, those measurements give the result of the lung function as one unit. They don't tell us whether different components of that unit function differently. And so what we did with lots of Jack's and John's time was we looked at the pig's lungs, which we scanned with the CT scanner. The figures here show a three-dimensional reconstruction of the pig's lungs. There's two of them, and there's 6 lobes illustrated with different colors. And these three-dimensional reconstructions come from slices of the lung, CT scans of slices, where you may or may not be able to see in the slide now some thin white lines that show the separation between the lung units or lobes. And these are then color-coded in the slice on the right, and these color-coded images are used to stack together the slices and reconstruct a three-dimensional representation. These scans of the lungs were performed twice for each animal. Once after the animal had breathed out, and that was one lung volume, smaller, and then some air was pushed into the lungs, like when we take a breath. And then when the lung was filled with the air of one breath, the ventilator was paused for about 20 seconds, and then a second scan was taken, and obviously this volume now was bigger. And the two volumes were used to measure how much air was going in and out of each of the 6 lobes. Now we have a volume that has gone into, say, the red lobe, and we want to know how much of that volume was oxygen. And so the oxygen percentage was calculated based on the oscillation in the partial pressure of oxygen in the airway. This was done with the bronchoscope. It's a device that is used clinically in these patients to look down the airway, down the mouth and the throat and the airways, to look inside the airways and the lungs. And there's a catheter, a hole inside the bronchoscope where the fiber fits nicely. And so we could direct the bronchoscope to the pipe, say, that enters each of these lobes, and measured the oxygen that was going in and was coming out. And those values in kilopascals on the vertical axis here are illustrated over a period of a breath of about 5 seconds in 2 different animals. One is the continuous lines and one is the dotted lines. And you see with the gray shaded area, we show the period of time during inspiration when air was going into the lungs, and expiration, which was longer, is on the white background. And so what we did was to, let me see, I think that this thing covers some of the equation here. To measure how much oxygen was taken up by each lobe, by each of these 6 lobes here, we used the tidal volume, the amount of air in liters or milliliters that entered that lobe and came out, and the difference in the oxygen partial pressure, which was then converted into percentage. So by having a volume and the percentage value, we were then able to calculate the volume of oxygen that was taken up by the lung. So say there was a higher amount of oxygen, volume of oxygen that entered the lung, a lower amount of oxygen that left the lung, and the difference was what the lung had taken up. And we could measure that amount of oxygen at the level of the individual lobes. And the results are illustrated here in panel E at the bottom right corner of the slide, where there's the oxygen uptake in milliliters per minute. It goes from 0 to 400. And the results are shown for 2 animals, C, which is a control pig, and SL is a pig following saline lavage. It is a reasonably common model of lung injury that simulates what happens in some patients in the intensive care unit with the usual limitations. What the results show, first of all, is that with this approach, we can detect differences in the overall oxygen uptake, in that the oxygen uptake of the second animal was greater than the first. But also, if, say, these 2 animals had some condition for which we needed to remove the red lobe in the lungs, you see that that red lobe does not contribute as much in the SL animal as it does in the C animal. So if we were to surgically remove this red lobe in the SL animal, we suggest that the function of the lung post-surgery would not be affected as much as it would if we were to remove the red lobe in the C animal. And we're currently looking for funds to translate this approach to patients. And I think that it seems reasonably logical that if we can quantify the contribution of the different components of the lung, we should be able to predict with a greater accuracy the post-operation lung function in these patients and hopefully contribute to their quality of life later on. Sorry. Next to last example refers to equine anesthesia. This is work that we did together with João Soares. I'm sorry, the name doesn't appear there. I'll move the controls higher up, at the University of California, Davis. And one thing that I didn't know at the time is that in a horse, the diaphragm, that's the main muscle that allows us to breathe, is not quite vertical in the horse. You see that it is at an angle and the gut is at the bottom. And so normally when the horse stands up, that doesn't pose, I imagine, any troubles. However, if you don't mind me swirling the animal around, if the horse needs to be anesthetized for some surgery and needs to go on their back, something which is called dorsal recumbency, you see that the diaphragm at this angle is now under the weight of the gut. As a consequence, the bowel's weight is on the diaphragm, which can collapse some of these lung regions over here. And I understand that this is a reasonably is a reasonable problem with ventilated anesthetized horses, where the management of ventilation is not simple. And so as an initial step, with João, thought of just testing whether this sensor can give a signal in horses, because we had never measured that before. And here we see the two arteries in the horse's mandible, where the one artery was cannulated to measure the arterial pressure and to take blood samples. That is a standard catheterization that is done for monitoring. And then a second one was used to insert our fiber optic sensor to measure the partial pressure or arterial oxygen. And we, as I said, we only wanted to see if the recording was possible, and we just performed a couple of ventilation maneuvers to see what results could be, what the response could be. And here we see when the airway pressure was increased in the lungs of these horses on the horizontal axis, it went from 0, which is the same as atmospheric pressure, 10, 20, and 30 centimeters of water. So at the end of expiration, we'd leave more and more air into the lungs of these horses. And then after a period of time, that pressure was released down to 20 centimeters of water. The period of time is around about 3 minutes at the top, and then a minute again on the right over there. And we see that here the normalized partial pressure of oxygen starts at 100, and then we see what happened in these 6 horses where each animal is represented by a different color. And you see that there was a reasonable variability in that for a couple of horses, while the airway pressure was increased, the arterial oxygen decreased. And that could be explained by a redistribution of the blood flow into the lung to regions that didn't receive as much oxygen. In contrast, for a couple of animals, there wasn't so much of a change. And for a couple of animals, there was a reasonable increase. And when pressure was released from 30 to 20 centimeters of water, you see again a similarly different response in that for 2 animals, the arterial oxygen went back to what it was. But for 2 of them, it increased by 4 or 5 times. So we think that this variability between horses is similar to what's reported in the literature. And we think that having something like this technology available for monitoring horses during anesthesia in dorsal recumbency could be helpful, because the veterinary anesthetist could tell straight away in a matter of a couple of minutes and see in real time whether the horse they're ventilating is the horse represented in red here, where this maneuver doesn't change much arterial oxygen at all, or if it is one that actually responds to this maneuver and for which the maneuver itself may be beneficial. And mind the importance of this is that in horses that are anesthetized, there's a higher risk of hypoxia. It's not enough oxygen getting to some of the muscle tissues where the weight of the horse compresses these tissues excessively for them to receive enough oxygen. And the very final slide is on work that is not ours, but it's from Dr. Whitauer and Professor Evans in Boston. It was published, I think it was initially online last year, but definitely it's an ACS Sensors 2021 paper. And the same technology was used, well, we have a leg of a man here showing some pain in yellow. The study really showed experiments in pigs, again, in a couple of pigs. The technology was applied to do some measurements related to compartment syndrome. Similar technology, it's a PPMA matrix, and again, using porphyrin. And you see that what the proposal here is that there is, the condition is one where the pressure inside the muscle is greater than normal. And that pressure inside the muscle essentially squeezes some of the blood vessels. And because those blood vessels become relatively occluded, the intramuscular oxygen levels are decreased. And so this group developed a sensor that doesn't respond rapidly. There's no need for it to respond rapidly. The changes in this compartment syndrome occur over a period of minutes, so there's no need for a rapidly responding sensor. But they coupled it with a mobile phone-based app and some LEDs and spectrophotometers that has potential to be a relatively cheaper solution to what we have. It's just slightly different applications. And I think it just shows the breadth of opportunities for fiber optic technology applications in the clinical context. And I think on this note, I'd like to conclude, and I'll thank you very much for your attention. And I think that we have a few minutes for questions and discussions, if you like.
Thank you, Dr. Federico, for a wonderful talk. I think there are a couple of questions in the chat box. I'll read out for you. Mm-hmm. So the first question is, the participant is saying, thank you for the very nice presentation. Since polymer coating were used and the sensors are used for continuous monitoring, did you check leaching of polymers from the sensor surface?
Okay, yeah. I'm just checking how many were used, and these sensors are used for continuous. Yeah, so continuous monitoring, yes and no. The sampling rate we use is 10 Hertz. So we get 10 data points per second. That is technically not continuous, but in the physiological and pathophysiological context we're interested in, it can essentially be considered as continuous. Did you check leaching of polymers for sensor surface? This is part of the technical work that Rongsheng led. I think the answer is yes. And in terms of we couldn't detect any changes over a period of at least 24 hours, if that gives a scale of what was done.
Okay. And this next question, nice presentation. An extended application query here. How can we translate these measurements in gas samples into liquid phase, such as non-Newtonian liquids, such as protein content in blood flow? And the follow-up question over there is, will the light scattering inside fiber coupler be sensitive enough to improve the signal strength for such fluids?
Yeah, that's a good question. So I'm trying to say, what we've done was gas and saline, like water and blood. Now, I'm not sure about the viscosity of these other fluids that Raghu may have in mind here. But in terms of the light source, one of the things that happened during our project was that the power of the LED light was increased rather substantially, and that allowed a greater strength in the signal. And that, I think, is one of the components of the system that one may want to look at if the signal is not sufficiently strong for their application in different fluids. Then trying to think of the latest example I showed from the Connor Evans group, the sensor was put into skeletal muscle. And in that sense, it's not quite a fluid, perhaps. It's not something that flows. And you can see they have presented some road traces in their paper, and it seems to me that the signal is clearly usable. It shows physiologically explainable values in the range that one would expect from those regions, whether the measurement was done just under the skin level or further down inside the muscle, further away from the environment, say. So yes, I'm afraid I don't have an exact answer for this question. It is a good question. I imagine there's two levels to answer. One is theoretical, based on equations that can be derived from the chemical physical properties of the fluids and the sensing matrices. And the other one, which is the one that I confess I like more, is the experimental one, going either on the bench or in vivo or in patients. All the calculations, do they actually work, or have we overlooked something for which in the end it doesn't actually work? Yeah. And then Nirmal, I see a couple of other questions. Can sensors...
Yes, so the third question is, like, can sensors be used for measurement of breath oxygen level, like the sensors which you have used?
Correct. So the short answer there is yes. Rongsheng and I put together a small paper where we used the sensor just outside the mouth. And that was just to demonstrate that we could measure the respiratory rate, how many breaths we can take per minute, which may be helpful in some neonatal intensive care unit where there are now novel approaches to measure the respiratory rate. It sounds like a very basic thing to measure, but it's not so easy to measure in practice. And the example I mentioned with the different components of the lung was actually from the fiber that was pushed into the bronchoscope, this pipe, and that has a little camera at the end, and you push it down the throat and drive it around the airways. And then where we found the location, the anatomical location where we wanted to measure oxygen in the air, the sensor was slightly pushed out of the fiber, and we recorded a few breaths. And when we had a few breaths, a reproducible signal, we then moved on to the next lobe and so on. So definitely, yes, it can be used in gas. One thought there is that the temperature of the gas of the air that we breathe in is typically colder than the air we breathe out. And if one's only interested in measuring the respiratory rate, that in a way doesn't matter. But if one wants to calculate the oxygen levels, may need to consider the temperature change and how that affects the signal.
Okay. Just following up on that part, so like, have you seen an effect of temperature on your sensors? Like, how much will it vary?
Yeah, we haven't studied very large temperature ranges, such as freezing and 100 degrees. I have to say the sensors we use are calibrated for using a temperature of 39 degrees, because in the preclinical stage, that is the core temperature of the pig's blood. But having used the temperature in those animals during the actual experiments could range between 37 and 40, say, vaguely speaking. And within that range, there wasn't a difference that was noticeable, at least. Yeah.
Okay, there's one more question. Wonderful presentation. Do you see any possibility where waveguide or chip-based sensors can be used as a better alternative? I think it's comparing with the optical fiber.
Yeah, yeah. So that is a good question also. I mean, there are several different options out there, and more are coming out. I think it depends a lot on the application. For example, say in the example where we measured oxygen in the airways with the lungs of the different lobes, technically speaking, there are other options. For example, we could use a fine tube that samples gas, and that gas can then be analyzed by infrared light or anything. Um, where I think that the fiber optic for our application is unique is in that I don't know of an alternative technology that can measure arterial oxygen at this high time resolution with able to detect rapid changes um, that could provide the signal that we need. So if we think back of the beginning of the presentation, ideally in that patient monitor, apart from the arterial blood pressure of the patient and their ECG that tells us about their heart and something else, we could also have, if we find that it is helpful, the arterial oxygen trace, which shows an oscillation that matches the ventilatory cycle. And that could be used if we know that the normal lung shows a given pattern, and then if we know that a diseased lung shows a different pattern, we could then use that signal to change the ventilation so that the ventilation is delivered in a way which is safe for any given patient at any given time. But yes, there's other approaches. I guess it all depends on what one wants to measure, what restrictions are out there, and what the best approach may be for a combination of reasons.
I think that's in the chat. So I have some couple of questions based on your answers. One was like in your presentation, you stressed more about the time response. Like, and even right now, you said that you got the in terms of time resolution, you got one of the best sensors that you have right now. So time is one parameter. So in terms of accuracy of the oxygen value, so how much percentage, or you'll comment on that, like what from the clinical point of view, what is needed?
Yeah, so very good points. One thing that I haven't mentioned actually is how fast is fast, and that's critical, because fast for somebody could be a few seconds. For us, I guess it's clear if a breath is 3 seconds, as I mentioned, we are looking at less than maybe 200 or 300 milliseconds for the response time, and that is what was achieved by the American, Austrian, German group. In 2002, and Rongsheng's sensors went down to less than 100 milliseconds as a response time. In terms of the accuracy, 2 parts of the answer. I imagine, clinically speaking, we'd want to find something which has less than 5% difference from the gold standard. In practice, at the moment, these sensors are still made in small batches. It's not produced in hundreds of thousands. So there can be some variability between sensors. That is reasonably minimal and can be controlled on the bench before the sensor is used. One of the limitations of this technology, to the best of my knowledge, is that if you imagine we have this blood vessel here, and we have the fiber that we insert into the blood vessel, it may be that the fiber sits right in the middle, but it may be that the fiber is stuck to one side or the other side, or it's bent, I don't think that or that it moves from 1 hour to the next one. There's not good reasons for which it should, but it could. And to the best of my knowledge, there's not a way that we can tell where exactly that sensing tip is. And if it is right in the middle of the blood vessel, the signal would be exactly the arterial oxygen level, plus minus whatever error. If it is on a side, at times we can tell, because if we alter something in the breathing where we should see a large and rapid change. If we don't see it, then it is possible that the sensor is not positioned correctly, and then we can adjust it. But in some ways, practically speaking, the error that we can measure on the bench may well be smaller than the error that could actually occur at the bedside, if that makes sense. So that is one of the limitations. The other one is that putting together the initial work and the different sections of the lung is that this sensor gives one value of arterial oxygen. Whether that value comes from two lungs which are essentially the same from each other and they're homogeneous across the lung, or whether they come from one lung that works and one lung that doesn't work, the sensor on its own cannot say. So that's where we combine different technologies. John did a big job on dual energy computed tomography, looking at how blood moved around the lungs. And Joba did the study with electrical impedance tomography, looking at how ventilation changes. So when you combine these technologies, then you can explain with a higher evidence base the arterial oxygen signal. Otherwise, there's quite a few assumptions that may lead to a potentially incorrect interpretation of the signal.
And then in one of these slides, you showed like there was a syringe pump type setup where you are withdrawing the blood and the sensors are on the side. So there are 2 points over here. One is, will like the flow of blood affect the response? And second was, you mentioned one place about the ethical part of putting the sensor inside the body. So can you throw some light for people over here who want to, like, if they are planning to take it to the next application, what ethical clarity?
Yes. So the second one is reasonably more straightforward than the first one. In terms of putting a fiber into a human's artery, there's a couple of things to be said. There's different types of fibers. And my understanding is that there's something which we call the silica, the glass fibers, which are relatively more brittle than polymer fibers. So if I'm the human that needs to have the first sensor put into one artery, I'd ask you to use a polymer fiber because if it's very unlikely that anything breaks. There's no leaching. That wouldn't be a concern for me. There's no toxic materials, and the pyrogenicity seems to be fine. But say for whatever reason, a mad individual comes in and hits me on the spot where the sensor is and it breaks, there could be a small bit of fiber that ends up distally in the blood. So if I put the sensor here and blood flows into my hand and a little bit of fiber breaks, it could end up in one of my small blood vessels here. And that would require a vascular surgeon, I guess, to take it out or something. Bear in mind, these sensors are meant to be used in patients who are mechanically ventilated and highly sedated, so they themselves wouldn't move. They have other catheters in other blood vessels anyway. But to reduce that risk, the intermediate step where the sensor sits just outside the body under positive pressure from blood that comes from inside the body is relatively safer, in that even if the sensor breaks, it's outside the body and there's positive pressure that pushes any broken fiber away from the patient's blood vessels. And in terms of the flow, in the animals, we typically have a cardiac output of, I think, 3-4 liters per minute. But I guess the best experiments there were done on the bench by John using a micro pump that could simulate that cardiac output and lower. And obviously, if the slower the flow, the slower the change. But the response time, the physical response time of the sensor itself wouldn't be altered, but it would be the time it takes for the change to take place that would be slower, right? And and that John's done by having a system of pipes where the sensor was positioned downstream of a Y connection. And there was a solenoid valve that switched between either of the two sources, where the oxygen levels were different. Say they would be whatever 10 and 100, and we used two different temperatures. So we had a very fast responding thermocouple where the response time was a few milliseconds, less than say 10, and that sensor was used to give us the response time of the setup. And then we looked at the response time of the sensor, to which we subtracted the response time of the setup, put it that way. So yeah, those would be the answers to the why do we put the sensor outside first, and what happens if the flow is reduced above a given flow. Then I imagine the response would always be the same, or the difference between responses would not be measurable, perhaps. Yeah.
Yeah, I think I'll just check if there are more questions in the chat. No, I'll just check on the YouTube live if there are any questions.
Oh, I see. Yeah.
So there's no question there.
Okay.
Okay. So Dr. Formenti, thank you very much for your kind talk, and it was a very good learning experience, and I'm sure it was same for all the participants who had joined today.
Yeah, thank you. And I'd say, typically when I listen to a talk, I don't have questions by the end, or I have few, I'm not sure I can answer them and so on. So if anyone has any questions later on, please do feel free to send me an email. I think it's in the flyer, Nirmal,
Yes.
or
Yes.
if you just Google my name. I mean, I get so many spam emails that it would be nice to get a genuine one on these. So yeah, feel free to send an email. Either I'll give the answer, or perhaps better, I'll ask my colleagues who know more than me what the answer is, and I'll share it with you.
Yeah, I've shared your profile, like your website link, so people can contact you from there.
Wonderful. Yeah. And yeah, thank you so much again, Nirmal, for organizing, and I'm glad it ran smoothly.
And thank you very much for your time. Yeah.
Enjoy your evening.
We'll connect again for some other opportunity on this.
Sure. Absolutely.
Okay.
Absolutely.
Thank you. Yeah.
Take care for now.
Have a great day. Bye.
And you. Bye-bye.